And I think, for me, it's been the best way to understand Darwin and Wallace. Right? Life varies from place to place in a highly non-random and predictable manner. That is because things have been evolving in the planet. If you were a creationist, the one way to refute evolution will say, well, I can find all these things all over the place. But if you trace the history of these things, you can clearly see that evolution, and not only the evolution of the organisms, but also the evolution, the life of the planet, is what has shaped these things. And there.
Yeah, my name is David. I'm the president of the Secular Society of MIT. We're a student group here at MIT whose mission is to provide a space for the humanist, agnostic, and atheist community on campus, and also to advocate for a number of important issues, among them matters like religious freedom, science communication, etc. I'd like to introduce our co-sponsor today, the Greater Boston Humanists, represented by Stuart.
Hi. We do exactly the same stuff that he's helping the campus organization do, which is we try to advocate for secularism, separation of church and state, the promotion of science, learning about and thinking about the world that we live in and our ethical responsibility, which is why we call ourselves humanists. We have a number of activities throughout the year. A lot of times they're in the Harvard area, but we have been one of the key organizations that started the Humanist Chaplaincy at Harvard, which has now been extended to the Humanist Chaplaincy at Harvard and MIT. And Greg Epstein is the chaplain, so if anybody knows any students who need any counseling and don't feel like the religious chaplains are their best bet for getting what they need, that's an option for people as well. And in addition, our website, bostonhumanists.org, is a link to our meetup page, which it is basically the meetup page that is our website. But we have links there to some of the other talks that we've had over the years, as well as upcoming activities. So I urge you to look into those. And we help sponsor the Darwin Day celebration every year. Darwin Day would be February 12th, the birthday of Charles Darwin, that we are here tonight to reference and enjoy with some science and some world travel, it seems. So if you have any questions, please see me, and thanks very much, David.
Okay, let me introduce our speaker briefly. So Dr. Gonzalo Giribet is the director of the Museum of Comparative Zoology, Alexander Agassiz Professor of Zoology in the Museum of Comparative Zoology, curator of Invertebrates and Molluscs, and professor in the Department of Organismic and Evolutionary Biology at Harvard University. He graduated from the Universitat de Barcelona, concentrating in Zoology and Genetics in 1993, where he also obtained his PhD in 1997, with one of the first PhD theses to use molecular phylogenetics in the zoology department. In 2000, he moved to Harvard as an assistant professor in the Department of Organismic and Evolutionary Biology, and curator in Invertebrate Zoology, receiving tenure and becoming first full professor in 2007. Dr. Giribet has received multiple internal recognitions from Harvard, including his naming as Harvard College Professor from 2017 to 2022, awarded every year to 5 professors across the university for their dedication to undergraduate education. Dr. Giribet's research focuses on the evolution and biogeography of invertebrate animals, focusing on arthropods and crustaceans, but spanning the entire animal tree of life, including both empirical and theoretical work. His main goal is to understand how animal diversity originates and is maintained, using whatever technique is needed. His work is deeply grounded in organismal biology, having conducted field work in over 40 countries, including all continents, including Antarctica. And that he has dived in all oceans, including the Arctic and Antarctic oceans. He's graduated 13 PhD students, many of whom now have their own research groups, and several MSc students. Dr. Giribet has authored or co-authored more than 400 scientific articles and 2 major invertebrate zoology textbooks. He currently serves as editor-in-chief of the journal Invertebrate Systematics and in the editorial board of about 10 journals. He has named 150 new taxa, including new species, genera, and families of multiple invertebrate phyla. Wow. In recognition of his work, 11 new species and 1 genus, Giribetia, have been named after him. Last but not least, Dr. Giribet's passions include windsurfing and wildlife photography. Without further ado,
thank you, David, for the nice introduction. It's a pleasure to be here. You know, I'm down the road or up the road, and I've never been actually in a lecture at MIT, so I feel like Good Will Hunting with all the formulas behind us. But it's really nice to be here, especially to celebrate, you know, Darwin's anniversary. And I wanted to start by, you know, giving 2 images that are very, very characteristic of Darwin, right? We've all seen the voyage of the Beagle, something that had inspired a lot of people, you know, to do this comparative biology by going to different places and comparing the biology, the animals and plants that live in these places, right? This is how we get to understand the process of evolution. And then you have one of the most characteristic images of Darwin with these famous finches. And I just want to tell you a little story first and then talk a little bit about my research. Darwin didn't really care that much about those finches. He put them all in a bag, didn't really know what island they were from. You know, I want to argue that Charles Darwin was really an invertebrate zoologist like myself, before he became famous for all the things. This is the end of his book on the origin of species. And this is something that not only is beautifully written, but it has a lot of things. You can dissect this sentence, and there's a lot of things there. There's like some being, you know, breathing into life, right? That could reconcile things like religious people, right? I remember when I was a kid, I was raised Catholic, and I will argue with the priest, well, I'm okay as far as you accept this. Yeah, you know, the creation is a metaphor, right? And you know, Darwin was taken as an anti-creationist and all that, and I'm not saying he wasn't. I'm agnostic, you know, or atheistic. But if you want to find also solace in a God, that doesn't mean you don't have to believe in evolution. I mean, these things are in the modern time, you know, it's, you know, there's different ways to approach it. And he left room for that in the end of his book, you know. But then he's talking about a spinning universe. And then, in the end, from so simple a beginning, right? From thinking about just simple cells, endless forms, right? That's what we have today. How life became what we can observe today, right? And to get there, you know, to evolve and give origin to all the forms that we see today, it spend a lot of time observing nature. Yeah? It's not something that you reach at the beginning of looking at these things. Everyone talks about the tree of life, you know, Darwin, evolution. He actually did not do a lot of illustrations. This is one of his schematic drawings that represents a tree of life, where he's trying to think about how species give origin to other species, or populations give origin to other populations. And in fact, this is the only illustration from the origin of species which is trying to also show how from certain ancestors, which they're converging, but they don't know exactly where, you know, you end up giving origin to the populations that by, you know, the process of evolution will end up giving origin to those endless forms. Okay? We associate Darwin with, you know, the tree of life and all that, but he really used that metaphor very little, yet he spent his entire time thinking about it. I want to portray Darwin as a little bit different than invertebrate zoologists. Here's Darwin with his barnacles. If he spent time working on one group of organisms, it was barnacles. He spent 8 years of his life making this beautiful, after the trip, sorry, I lost one slide. Maybe it'll come out later. But spent a lot of time after his voyages working on these beautiful, well, or not so beautiful for some people, barnacles, and the gooseneck barnacles and the other ones. And he had these monographs, multiple monographs, where he described new species, looked at their development, spent 8 years comparing these things that he had been collecting around the world. And of course, the thing that made a difference for Darwin was the opportunity that he had to travel the world for several years and observing everything related to biology and geology around the world. Right? It's very difficult to reach the conclusions that Darwin reached if you just stayed in one place observing your surroundings. And really, these things play a very important role. I have to say 2 things. One is that, since I was a kid, I was inspired by this travel. I found today, I was looking, these are my first 2 books on Darwin. They're in Spanish. I was a kid. These are when I was 14 years old, 2 of my favorite gifts. The Origin, Illustrated Origin and commented by Richard Leakey, who I had the pleasure of meeting in Kenya a couple years ago, and Alan Moorhead's narration of the Voyage of the Beagle. So these were, my favorite books, and of course, since then, I was a kid, interested in evolution. Since then, I've got to read a lot more things, obviously, from Darwin and about Darwin. And this has inspired my work, right? That Voyage of the Beagle, now it's very different. We don't have to go 5 years around the world. We have planes and we can go back and forth. But today I did a search in our database in the Museum of Comparative Zoology for specimens collected in my expeditions, and then you can generate a heat map of the areas that where I've collected the most specimens, right? Each point in this heat map is one collecting event, one specimen or one group of specimens, and in total is about 10,000 lots of specimens that are now in the permanent collections of the Museum of Comparative Zoology from the work that I've been doing around the world, trying to study evolution. I mean, my main goal is to compare the biotas, the animals, the invertebrates that live in different continents, for example, to explain things like the breakup of Pangaea, right? The breakup of Gondwana. When did the Atlantic Ocean open? And those things left traces in the animals that live there, in the genomes of the organisms that live there. And in order to study that, we often need to go to all the different points in the map to find these groups of organisms that are around the globe, and then bring them back into the lab and do normally the genetic comparisons to see what they do. Here's a little bit more detail of the map. I've done a lot of work in New Zealand. This is one of the most intensely collected areas. Like me, obviously in Southern Europe, because when I grew up, I actually had a collection before coming here of several hundreds or thousands of molluscs that now they are in the collections of the Museum of Comparative Zoology, and done work in, all continents, all oceans, a little bit in the northern Greenland, a bit in Antarctica. I'm actually going back to Greenland next summer. So, I get going back to some of these places, among other things, because the techniques for, what we can do today in genomics has changed. And I have to go back to places where I collected things maybe 20 years ago. It's like, I wish I had collected them with RNA later, or I had frozen specimens or things like that. But Charles Darwin had many other output that is not just these big books that made him really famous. This is actually one of his scientific papers many years before the origins that I read for my research. This is a paper on a weird animal phylum. A lot of my work is trying to understand the relationships between the animal phyla, the major lineages of animals. We understand for phylum a group of organisms that shares a ground plan. For example, chordates is a phylum, they share the presence of a notochord, or sponges is a phylum that shares the presence of special types of cells called squamatocytes. We have arthropods, we have molluscs, we have 33 animal phyla. And one that is a bit obscure and small are chaetognaths, the arrowworms, these mysterious pelagic animals that have, they're incredible predators. And that for a long time, people have been trying to figure out what are they related to, because every anatomical character that they have is different from that of other animals. Well, Darwin already noted that. The first sentence of this paper says the species of this genus, Spadella, are remarkable from simplicity of their structure, the obscurity of their affinities, and from the abundant and infinite numbers over the intratropical and temperate seas. So he already talks about the obscurity of their affinities, and later on at the end he concludes by saying, I will conclude by expressing a hope that these few observations on the propagation of this curious genus might add more competent judges than myself in ascertaining the true affinities, right? So we're talking about 1844, he wrote this paper on the affinities of chaetognaths. And you might think, well, people will resolve that quickly, but in fact, they didn't. This is one of those animal groups that until very recently, we have been struggling to understand what they are, as Darwin predicted already, in 1844. This is a paper in 1993, beginning to use some modern techniques, and at that time they postulated that those chaetognaths, maybe they're related to deuterostomes. We are deuterostomes, echinoderms are deuterostomes, or maybe they are much more basal with that question mark on the animal tree, and that's a highly simplified animal tree. And if you actually read different papers that people have written on chaetognaths, they feel related to deuterostomes or the ascalminthes, for example, nematodes like the roundworms or things like that. It's just sister group to all other protostomes, which is the major branch of bilateral animals, or related to ecdysozoans or arthropods. So basically, they've been bouncing around the tree of life for a long, long time. And only very recently, since there was a paper in 2019 that anchored them with genomic data. So this is the first time a paper that used some signal from Hox genes and entire genomes to finally anchor these chaetognaths together with things like the wheel animals, the rotifers, for example. And so those predictions from Darwin, that it will take a long time to solve these things. He was a good invertebrate zoologist, and yes, it took a long time. But finally, I think we solved that. And that's some of the type of work that we're doing in my lab, trying to pinpoint a lot of these weird animals that it's been very difficult to figure out how do they relate to other organisms. Here are those pleasure articles, so we really found pleasure in understanding rare animals and what is more strange than barnacles, which are crustaceans related to shrimp, but at the time people thought they were mollusks related to clams and snails. And only by understanding the anatomy of the larvae and then the adult anatomy, that they have appendages, so those shell plates that are a form of calcium carbonate, they're not related to the ones of mollusks. This is an innovation of these things. And these are some of the plates from his monographs, where he really spent all that time studying barnacles. This is one of the few of Charles Darwin's specimens that are here in our very Cambridge, Massachusetts. We have a few in the MCC, very few. This is, for example, one of his echinoderms that collected during the voyage of the Beagle in Patagonia. And again, most of the specimens that we have, they are invertebrate animals that he found fascinating, that he collected. This was one of them. It's in our collection of invertebrate paleontology in the MCC. So with this part, I want to argue that he was fascinated by all forms of life. Of course, he spent the latter part of his life with breeding pigeons or studying things that he could study in England. But when he was around the world, he really was captivated by all the diversity, especially rare and strange animals. Darwin without Wallace, they were both co-discoverers of the theory of evolution and very remain good friends. There's rumors that because Charles Darwin published his big book before, even though they presented the paper together, then Darwin published his book, that Wallace had been resentful. Wallace only admired Darwin. In fact, he even coined a term for evolution called Darwinism, not Darwinism. And he dedicated one of his major books, this book on the Malay archipelago, to Charles Darwin. He was also a pioneer, and he really put a lot of emphasis in geography, right? The laws of distribution can only be arrived at by comparative study of the different groups of animals. The laws of distribution, right? Why do we find these animals here and not there, for example? For this study, we require a common system of regions and a common nomenclature. So he's trying to establish the biomic regions of the world and use those to compare, like the neotropics with the Indo-tropics and things like that. And so geography played a big role in Darwin's voyage, obviously in Wallace's research, and entered the world of biogeography. So what's wrong from this picture? And don't tell me penguins don't drink coffee. Imagine that. What is wrong from a biogeographic point of view in this picture?
Penguins and white bears are not.
They're not in the same place, exactly, right? So that's what Wallace was talking about, right? This is, I don't care if they talk, I don't care if they drink coke, they cannot coexist. And the reason why they cannot coexist is because they evolved in different parts of the region. They could probably live in the same environments. They're adapted to those ecologies of cold places with no light in the winter. But the bears evolved in the northern hemisphere. They did not traverse the equator, and penguins evolved in the southern hemisphere. And yes, they live in Galapagos. I know you all know that. But Galapagos is only the very cold front of the Antarctic region, right? They really can't cross a warm ocean, right? And that's what we're studying in biogeography. Understanding how animals achieve their distributions, and we integrate evolution with the history of the planet, with the ecology of the planet. That is what really motivates me, and I think for me it's been the best way to understand Darwin and Wallace, right? Life varies from place to place in a highly non-random and predictable manner. That is because things have been evolving in the planet. If you were a creationist, the one way to refute evolution will say, well, I can find all these things all over the place. But if you trace the history of these things, you can clearly see that evolution, and not only the evolution of the organisms, but also the evolution, the life of the planet, is what has shaped these things. And there are three disciplines that come together in the field of biogeography. One is understanding geology and earth history. And I confess that when I was a student, I didn't like geology very much. Maybe our professor in geology wasn't that good. I wish I had known more geology. I'm trying to learn, but it's really a fundamental component to study evolution in general. Then there is ecology, right? Yeah, the penguins and the polar bears could live in the same area. It's just they won't, because they've never been exposed. They can't cross the equator, and obviously evolution, which links all together. So one of my favorite plants, I'm not a plant biologist, but sometimes, there's people who like plants, cycads, and they have that distribution. I always tell my students, it's great when you give a talk, maps and photos of organisms, they make it interesting. This is the modern distribution of cycads, right? And we can expand that distribution in many, many different ways, One of them, obviously, and this is what they had at the time, they thought the planet hadn't moved, or the continents hadn't moved, right? And to explain a distribution like this, you could say that these things are able to disperse over oceanic barriers, or that they lived everywhere and they have gone extinct in some places. But those are the explanations that they had at the time, right? And in modern biogeography, or you could speculate, these things lived all over Australia when the climate was different, because they knew that climates had changed through time by geological study, and now because it's become arid, it has disappeared from the scene. So you can always use ecological arguments to explain how distributions of these organisms have changed. But they were missing one big piece. And that big piece of the picture is something that was introduced much later and discovered or proposed by this German meteorologist, Alfred Wegener. The idea of plate tectonics. The idea that continents were not, fixed on land, but they were moving. They were reconfiguring completely those different land masses, right? And he proposed this theory of continental move, of plate tectonics, that actually wasn't really accepted, because even though he had interpreted that based on geology and other aspects, he didn't really have the mechanism to explain how continents move. So the idea that continents moved actually wasn't only from Wegener. It's an old idea. There's this Italian scientist, Snyder Pellegrini, that in 1858 they already recognized that South America and Africa kind of fit together, and he proposed this theory, right? That there had been the big deluge that had separated those continents, and then that's what we have the modern Earth, right? So the theory of continents changing had existed. Wegener really proposed a much more solid theory, but there was no mechanism. And without a mechanism, the scientific community was not ready to accept that. His model, was based on evidence from geology. For example, that you will find tillites, these, rocks from volcanic activity that were lined perfectly between South Africa and parts of South America. The presence of the same fossils across these very distant oceanic openings today, right? So he did have an explanation for how these things happen. He even proposed a model how at different times the continents have had different shapes. Okay? But again, this was only, if you want, by drawing things on a board and moving them around. They didn't know, he couldn't propose the way to do that. And it was actually the Cold War that gave us the tools to understand what's happening in the bottom of the oceans. And a lot of the deep sea exploration happened to, basically go between Russia and the US, with submarines and exploring a lot of the ocean bottoms. When people discover that there are parts of the ocean that are extremely young, that are being formed constantly, and there are parts of the ocean that are very old, that are being subducted on their continents, and that these movements of plates, the constant generation of new ocean floor in the middle of these oceanic cracks, right? The mid-ocean ridges, this is forming, new ocean crust that keeps expanding, and as it grows, it separates the oceans. Obviously, it's very difficult to predict how things happen, because you can see South America is being pushed from here and here. But the ocean floor is much younger here, meaning that it's spreading much faster. So even though it had opened from the Atlantic, now it's being pushed in the other direction, right? Most of the ocean, well, we know that land can be very old. There are rocks that are billions of years old. The ocean is extremely young. And in fact, most of the ocean in the world is not older than 100 million years. Only the Tethys Sea, in the middle of the Mediterranean, you have ocean basins, ocean rocks that are around 240 million years. So this is an extremely dynamic system. Then in 100 million years, these oceans are changing. We know a little bit that the Himalayas are formed because basically India broke up from Antarctica, and in a very short amount of time, they shoot off north with so much energy that formed the Himalayas, for example, right? It keeps pushing against the north with the formation of some of these ocean floor. You have very fast spreading, ocean floor here, for example, right? And as it gets against the continent, because the ocean floor is lighter than the continent, it goes down, right, and gets resorbed by the crust. So this model of the movement of the oceanic plates, the birth and expansion of the oceanic plates, is what today allows us to understand what's happened to these biotas during all that time. I wish Darwin had known this, for example. Is that fiction? I mean, you can see in places, the Rift Valley. There is in the east of Africa, the Rift Valley is just forming a divide. That one day will fill up with water and it will separate, right? I mean, some of these processes are happening today. And we have this model where ocean crust is being formed, where continents are breaking apart, and this is what we're trying to use to understand the distributions of the organisms today. So the second part of the talk is going to be less, I want to talk a little bit about the research we do with one of the most fascinating animals that lives today. I didn't have a video because I didn't know if my network was going to work at MIT. I'm sure that you guys don't like Harvard people to connect to your network. So, but if you Google, when you're home, velvet worm hunting, there's spectacular videos how these animals shoot this glue to trap insects. And we do a lot of work on obscure animals that live in the leaf litter, in the soil, in humid environments, in rainforests, because they don't tolerate changes, they don't tolerate desiccation, means that they are put in their forest for hundreds of millions of years. In fact, we have fossils of these velvet worms that look exactly like today that are 300 million years old, right? So when all the continents look very different, these things were already everywhere. And since then, they've been writing on the different plates of the continents, and they're telling us, they're reading us back the history of how those continents separated. And we use velvet worms, but we use some other animals to study these things. This is one that we collected this past January in St. Vincent and the Grenadines. And velvet worms divide into two families. A tropical family you have here in green, we call Peripatidae, with one species in West Africa, about 3-4 species in Southeast Asia, and a large radiation of more than 100 species in the neotropics, in the Americas. And then we have another family that we call Temperate Gondwanan, which is in southern South America, it's in South Africa, Australia, New Zealand, and New Guinea. And these two families have parallel but different evolutionary histories. One of the things that I find fascinating as a biogeographer is that there are connections across continents, for example, in these ones, right? And we know that those continents were once together and that they've been breaking apart. So they've maintained that connection. The same thing with the green ones. But they haven't traversed north to south, either here or here, right? Here is a bit more complex. But there's always been contiguous land between southern Africa and Central Africa. And there's always been contiguous land between South America and southern South America and northern South America. And many groups of organisms haven't walked this distance, but they have expanded with the breakup of the continent. This is to tell you how put they stay. So a lot of what we see here, we expect that is a result of evolution, of clinogenesis, of how as the landmasses break up, these animals just go into the other landmass and they keep evolving there, but at micro scales. And so this is some of the work we do. I mentioned there's a fossil from 300 million years ago. That fossil is from France, where they don't live today, but 300 million years ago, France is not what it is today. And there's one more recent fossil that you have there on the right, that's from that famous Burmese amber from Myanmar. This is Cretaceous amber, 100 million years ago. There were dinosaurs walking around, and those things looked exactly the same as they do today. So we studied visit. We go to all the places where they live, all those different places in the continent. We bring the specimens back to the lab, we sequence a few genes at the beginning. This is a few genes. And we find that, yeah, that our phylogenetic reconstructions, when we compare the DNA of these animals, they separate between the tropical family Peripatidae and the circumantarctic family Peripatopsidae. And if we look at the details, what we find within these two families, it's not that these things are randomly distributed. They're highly correlated. So, for example, with the family Peripatopsidae, the first few species that you have in the top correspond to West Gondwana. So southern South America and Africa, they work together for a longer time. And all the rest correspond to East Gondwana, Australia, New Zealand, New Guinea. And even if we pay attention to some of those things, today we find that all these species are found in New Zealand and Tasmania. These other species here are found also in New Zealand and Tasmania. And as you go up in the tree, some are found in Eastern Australia, some of them in Western Australia. So even today at that level, they have very strong biogeographical structure. And that's why we choose these species. A lot of people ask me, why don't you work on insects? Insects fly around. They're not going to keep these hundreds of millions of years history because they move around too much. Okay. So the first study that we did, that's based on, basic sequencing. This is an old study now with sequencing a few genes using Sanger sequencing with PCR amplification. A few years ago, we had to explain what PCR is. After COVID, everyone has heard of PCR, so no need to. But it's only a few markers. We wanted to test that with more genetic data, right? And one of my grad students did this other study where we actually had to recollect a bunch of these samples, preserve them in RNA later to extract total RNA instead of DNA. The RNA is less stable. And with that total RNA, we had mRNA libraries and sequenced the transcriptomes of those animals. Basically, most of the genes that are being expressed in those animals were able to sequence, and now instead of having 4 or 5 PCR amplified markers, we had a snip of the genome, and we could use 1,000 genes to resolve these phylogenetics. And the results that we got were very similar, to the ones that we had before. Very similar results. For example, also split between East and West Gondwana in the top. That's the family Peripatopsidae. But we found that one of the groups that we were interested in, this radiation of the neotropics, was very poorly resolved. Meaning that there's no support for a lot of these nodes, there's no resolution for a lot of these nodes. So if we wanted to resolve these radiation of the neotropics, this is something that probably happened during a short evolutionary time frame, we needed to look at other types of genetic markers. And one of the things that we wanted to do is, oh, by the way, this is a specimen that we collected in Trinidad a few years ago with 2 of my colleagues who are sitting in the back here. They were in this field trip. This is a very large, one of the largest villa worms. They're about this size. We put it in a box, and the next morning we had a baby. This is a just born baby. In fact, they had to. These animals are viviparous. Some of them have placental type of reproduction, like mammals. Others have internal. The eggs basically eclode inside, and some of the species from Australia and Tasmania, they lay eggs, so they have a huge variation in reproductive mode, even though they all look very similar. So what we wanted to do then is use some of the newer techniques now to do genome representation from more specimens. I mentioned earlier that we needed to go back to some of these places to collect fresh animals, to extract RNA. But our museums and our collections are full of specimens in ethanol that have been collected for sometimes hundreds of years. So we turn to other techniques that are based on hybridization of highly fragmented DNA, that you can develop probes that are specific for those, 1,000 genes, for example, and then those probes will hybridize against your sheer DNA, and then you can bioinformatically reconstruct a bunch of markers, and then you will compare among species to do this phylogeny. So we could use now a lot of the other specimens that we had in collections, but one of the things that we needed to do in order to develop a probe set for these UCEs, these ultra-conserved elements, was to do the, I'm going to skip this one in the interest of time, to sequence the first genome of these featherworms. And this is something that actually proved pretty difficult. The genome that we sequenced had 6 gigabases. This is more than twice the size of human genomes. For something that we had no reference for before, so assembling it was complicated. From the entire genome, only 29% are the featherworm genes, if you want. And the other 71% are all types of transposons, repetitive elements, all of these things that are a nightmare for those who work on genomics. Maybe they have some meaning, we haven't really looked at those things. But in the end, we were able to assemble a genome for these things and then use the non-repetitive sequences to develop probes to capture a much more diverse pool of these featherworms. So what we're doing now in the lab are larger phylogenies. This is very similar to the one that we saw earlier, but instead of being based on 4 standard markers, this one is based on a matrix that contains about 1,400 genes, okay, and all of them. So we developed this probe set. This is part of the research that former grad students showed us how to do. This paper has just been submitted, or is being submitted this month, and we hope that we can continue to study these things. We find very similar patterns to what we saw earlier. The purple family here is the temperate Gondwana 1. We find the division between West Gondwana and East Gondwana. We have Eastern Australia monophyletic. We have Western Australia monophyletic. So these things are really, you don't have one single species that goes with the Western Australia clade in the other place, right? And then in the family, in the green family, Peripatidae, we found some of the results that we had seen before. Also, a split between East Gondwana, in this case are the ones from the Neotropics and one species from Gabon, and the only few specimens that we had from Southeast Asia. So that division was there, except that the arrow shows us a specimen that we were expecting to fall here, but it goes earlier, like an older lineage. And when we saw that first, I said, well, there must be an error here. This is a specimen that was collected in Guyana, and we then resequenced it. We went back, we only had one specimen, we went back, resequenced it, and both of the sequences came in the same place. So that was a very unexpected thing. We can also find unexpected results, obviously. And when we looked at that animal, this is a typical little worm that I was showing you earlier. And when you look at these yellowish structures on the dermis, those are called primary papillae. The primary papillae look like the one up there, you know, like a structure with a mid piece and like an apical piece. And actually, when we looked at our specimen from, and you can even see them with your naked eye there, all these bumpy things, you know, each one of these things, these are the apical pieces that stick out, really visible, and they give us the aspect of the lines, the form, the cuticle of these animals. But when we study the one from Guyana, you know, we see that there are some of these yellow dots there. When we look at the scanning electron microscope, we don't really see a distinction between primary and secondary papillae, which exists in all the other featherworms, and then they give them these, you know, annulated cuticle aspect, right? So these ones don't have that annulated cuticle. They don't have, when we look at the scanning electron microscopy, they don't have the expected primary papillae. They have some papillae with a tiny mid piece and something that looks like the apical piece, but it looks very different morphologically. It looks, you know, it appears in a weird position phylogenetically. So a lot of this work of looking for these things all over the place also allows us to discover new lineages and lineages that might have different evolutionary histories. So that's a little bit what we do, and you know, some of the projects that we have today is, for example, to understand a radiation of these velvet worms in all the Caribbean islands. These are all the species that are distributed. Basically, every little island in the Caribbean has a species. It's very unfortunate because we have to go to all those islands. We've got the ones in yellow already collected, and we have, the ones in red already collected. We need to go to the ones in yellow, so you know, that's what we do. That's why I was in January doing some of these field work. and we're very privileged that this work that is, I think, very interesting from an evolutionary point of view takes us to all these fantastic places. And some of these places we go, it's not easy. For example, this is the forest, the rainforest in Gabon. We went there a few years ago, also with my colleague sitting in the back here, Ligia, and we were looking after a species that had been collected only 100 years ago, right? So these things, no one has seen some of these animals in 100 years. We had some old names that were actually missions from that part of Africa, and we were able to survey these things and spend 3 weeks looking for this animal. And the last day of fieldwork, we found 1 specimen. So I know that all this looks very charming and all that, but being 3 weeks looking for something and finding only 1 the last day generates a lot of stress, especially if people's projects depend on those samples. The one that I just showed you from that nice photo of the big one from St. Vincent, that was the first velvet worm ever discovered. Was discovered in 1823. It was then collected once in 1892 and once in 1984. I spent 5 years trying to get the collecting permits to get to St. Vincent, and finally we were able to go there in January and collect some of those specimens. They're all not that difficult. Some of them are in places that are easier and more accessible. New Zealand, it's a beautiful place to do field work. We were just there in one of my many trips to New Zealand with one of my grad students is working on the animals from there. We were able to collect lots of specimens. These are actually 2 different species there's 3 individuals that belong to 2 species from a single locality. You know, this is the one that lays, those are viviparous. This one is one of the ones that lays eggs. So we're also trying to understand the different biologies. Why do they live in different environments with these different biologies? But I just love them, so I'll show you some images. These are from Tasmania. We were there January last year, also collecting a bunch of these beautiful, and they all look very similar. There's not a lot of anatomical characters to distinguish them, and that's what we do a lot of the genetics. You can see my students and postdocs here having a hard day in this place collecting, and we spend a lot of time in the floor, you know, looking in the leaf litter, breaking rotten logs, looking for these really small things. We do that not only with velvet worms, we do that with other groups of animals. Like, for example, these are some of our daddy longlegs. A lot of the research in my lab is in daddy longlegs. I had to show them, or with Rhizinulae, and they're always these groups that have this chunk distribution, right? We have a genus that lives in West Africa, two genera that live in the Americas, and we're trying to figure out, are the genetic data of these things consistent with a breakup, an opening of the Atlantic Ocean? And we, in all these groups, have to say that we find a lot of that signal. So it's work that I think puts a lot of these animals into a much broader context. It's work that is really fun. I love the animals. I love the places where they live. And with that, I just want to thank you all for coming here. Sohan for inviting me, except that I just learned that he's not here anymore. But thank you, David, as well. I hadn't prepared this slide with your name. And to Charles Darwin for making us think about all these things. I don't know if there's any questions, but thank you very much.
Okay, let's do a Q&A. So if you have any questions, just like, I guess, feel free to ask them right now. Yep.
Professor, you mentioned that the evolution of the velvet worms the cell diversification begins in Jurassic period. And so this is a really long history, and I guess many of the shell-code genes might have lost during the diversification, and maybe some genes have experienced a back mutation. And did you encounter this kind of challenges? I believe this might also both of them influence how the phylogenetic looks like, and how you deal with it.
Yeah, so we use phylogenetic methods that are very standard today to study, all groups, for example, right? So there's different models that have to do with the age of the divergences, the type of data we select also, right? If you select very fast evolving genes, there's going to be a lot of back mutations. So one of the choices of these ultra-conserved elements is that the core of those genes is highly conserved across the entire group that we're studying, right? And we can select, if we want to go far from the core of the UCEs to things that are more variable, for example, to study population level processes, right? So we can control what we're using. Here we're trying to use the more conserved region to answer questions that are related to Jurassic, divergences. But if we wanted to look at recent speciation, we will take the flanking regions, which are more variable.
Thank you.
I'm curious about one thing, historically. On the earlier slide, you indicated where Russell wanted a nomenclature and classification system. Presumably something of the sort was already in existence. But how did that come about after Russell identified the need for clarity?
Yeah, I mean, that part is a bit stranger to me. There was a system in place, the Linnean system, right? Now, the Linnean system was based on, Linnaeus was a creationist, and it was based on similarity, right? And I think that in that sense, Wallace probably thought that we needed a system that was based on evolution. But the reality is that evolution generates a lot of similarity, right? There are also convergences and things like that. And just an anecdote, for example, Wallace visited Cambridge. Wallace was a, polymath. He did all sorts of things, scientific and non-scientific. One of his big things was like spiritism, right? And he was out there. One of his books is on life on Mars, and one of his passions was museums. And he did a tour to visit American museums. And he came here to the NCZ, to our own NCZ, which, if you know, was founded by Louis Agassiz. Louis Agassiz was one of the fiercest enemies of Darwin, and he was a creationist, among other bad things that he did. He had good things and really bad things. Okay? So Louis Agassiz had the NCZ, he founded the NCZ, and when Wallace came here, they said, wow, in the land of Darwin, our National Museum, the Natural History Museum in London, just has these random things of a bunch of birds and a bunch of things. But when I entered the Museum of Comparative Zoology, which was actually created by the fiercest anti-evolutionists at the time, he found evolution because he organized things by biogeographical regions and things like that. So it's very hard to ignore evolution, even if you try. So what I mean is that the Linnean system had a lot of things that weren't really good,